Namaste, Dr. Sangeeta.
Yes, namaste.
Yeah, namaste. You please try to share your PPT which you want to present so that we can check whether it is coming properly or not.
Yeah, sure.
कुर्सी है. Okay, can you bring
Yeah.
it to the presentation mode?
Yeah.
Okay, just move slides. Okay, okay. Yeah, it's moving
Okay.
properly. Yes.
Okay. Okay. Thank you.
So your voice is also clear. So we will start sharp 2:30, just 2 minutes more.
Sure, sure.
Namaste, Dr. Bhavana.
Namaste.
Yes, madam. Welcome, madam. We
Namaste.
will,
Dr. Mohan भी हैं?
yes, ma'am. Hello.
Hello.
हाँ, madam. We will start in just two more minutes, madam.
No, no, I have switched off the speaker. Because I also switched off
Actually, if the two devices are there in the same place, then there will be echo of the voice. So one speaker you have to put off.
हाँ, नमस्कार डॉक्टर मोहन.
Madam, Dr. Mohan, sir, है क्या? Okay. Mohan sir is today in Delhi, madam.
Oh, okay, okay, no problem.
Yes, madam. Yes, madam.
Incidentally, I was also in IR Delhi today in the first half where for a CME.
Yes, madam. Yes, madam. Sir informed that madam is here only.
Yeah. Academic activities, full of academic activities today.
Yes, madam. Full
Yes,
tight schedule. Yes, madam.
great.
Namaste, everyone. I welcome once again in this. Transnational research lecture series, and this topic, that is genomic science, this will be dealt under the guidance of Dr. Bhavana Parashar madam, and there will be series of lectures of this particular topic. So to formally introduce Dr. Bhavana Parashar, I will request Dr. Nikita to please introduce our guest speaker and the guide.
Good afternoon. I, Vaidya Nikita Naik, welcome all for the transitional series. Today's topic is genomic science, which will be conducted by Dr. K. Sangeeta, PhD scholar, ACSIR, under the guidance of Dr. Bhavana Parashar Madam. I take opportunity to introduce even Bhavana Parashar Madam. She is a distinguished scholar and a researcher, is an alumni of the University of Bombay and Gujarat Ayurveda University, Jamnagar for over 25 years. She has advanced Ayurveda through her work at CSIR, where she serves as a senior principal scientist, principal investigator of Ayush Center of Excellence of Ayurgenomic at CSIR IGBI, New Delhi, and professor at ACSIR. A pioneer of Ayurgenomic, she has provided genomic and molecular basis to Prakriti and Tridosha, discovered predictive health markers, and developed AI ML based algorithms for Prakriti analysis. Madam has contributed to NCSN's Desh Ka Prakriti Parikshan Abhiyan and Rashtriya Prakriti Parikshan Karyakram, enabling personalized preventive health at the public health level. Wisely published with nearly 30 peer-reviewed research papers, she has integrated Ayurgenomics into academia, trained future scholars, contributed to global Ayurveda standards, and delivered lectures at national and international forums, including the WHO. In recognition of her outstanding contribution, the Government of India has honored Bhavna Madam with the National Dhanvantari Ayurveda Award. I request you, Dr. Sangeeta Madam, to start with the presentation, please.
Thank you for the introduction. Thank you for the introduction, Dr. Naik. And I think today's lecture on Introduction to Genomics, I hand over to Dr. Sangeeta, who will take it for you today. Dr. Sangeeta, please start.
Am I audible?
Yes, audible.
Yeah.
The
Okay.
PPT is also visible.
Okay. I'll just hide this. Okay. Yeah. Good afternoon. Welcome to today's session. So today the topic is Introduction to Genomics, and I'm Dr. Sangeeta. I'm a PhD scholar. I've done my BMS and I've done my MD. After that, I've joined PhD under Dr. Bhavana ma'am in CSR IJB, which is an interdisciplinary institute. And currently, I'm in my 5th year of PhD. So now we'll start the session. So the today's highlights that we will go through. So we all have studied genomics, genetics, DNA, everything in our 12th standard. Afterwards, we came to the medicine. I would like to kind of bridge what genomics is for you in this lecture. So starting with what is genetics and what is genomics and the core difference between it. Second, basically the DNA and the RNA, the life in a spiral, decoding them. Third, we'll go to the central dogma, the one-way street of molecular biology, which is it was laid down by the Crick, and how the central dogma came into being and what are its exceptions. Next, about the genes. So we have all heard of genes and how it is an integral part of DNA. So all about the genes, the functional unit of heredity, and at the end, the different projects that have been taken up in this area, which have led to shape the genomics that we know today. So to revisit, we all know the basics that we have cell, and inside the cell, there is nucleus. Inside the nucleus, we have chromosomes. Chromosomes is made up of chromatins, which is in turn made up of DNA. Now, specific sequences in DNA are called as genes, and genes have, in turn, as you can see in the figure below, they have got coding and non-coding regions called exons and introns. So, with this introduction, I will move on to we have all heard genetics and we have all heard genomics. Many of us might be confused as to what is the difference between the two. So I would like to take you through both the terminologies and its evolution along the time frame so that you can have a better idea about what these two departments mean. So we all know the laws of inheritance. So in pursuit of knowing the inheritance laws, Gregor Mendel in the 19th century conducted experiments, and it came into the conclusion that single genes control single traits. So that led to the advent of genetics. It deals with how specific genes influence traits and how they are passed from one generation to the next. After that, in 1953, as we all know, there was the discovery of the structure of DNA. And afterwards, there was powerful sequencing technologies that were found out in the late 20th century, with the advent of which genomics came into being. So genomics is basically the whole genome, not just one gene. So the whole genome functions as a network rather than as an isolated gene. So if you look at the core difference between both the departments, So genomics is basically the study of the entire genome, the full set of DNA, including all genes, its non-coding regions, the regulatory elements, as well as how they interact with each other and with the environment. And genetics is basically the study of individual genes, their structure, function, inheritance patterns, and their role in different traits and different diseases. Now, what is the level of study in both that we have to see? So genomics is studied in a genome-wide scale. So it looks at different networks of genes, different gene-gene interactions, what are the impact of it, gene-environment interactions, then multiple regulatory sequences, structural organizations, and their collective effects. Now, genetics is mainly gene-centric, examines different sequences of genes, different mutations that occur in the genes, and the direct phenotypic effects. So basically the structure, function, inheritance of the genes, and expression of specific genes and their variants. Now to do both the genomics and genetics, the approach that is taken in genomics, it is basically, we would have heard of sequencing methods. So it involves high throughput sequencing technologies like NGS. Then there are genome-wide association studies, different bioinformatics methods, and systems biology approach. For genetics, it's the classical Mendelian approaches like gene linkage studies, candidate gene studies, PCR, that is polymerase chain reaction, Sanger sequencing for specific loci of the genes. Now, where is this important? Genomics is basically important because it is a very pioneering area for personalized medicine, as we all know, for disease risk prediction, for predicting the risk of different diseases at the early outset, and genome editing. We all have heard of CRISPR genome editing and other new technologies that are coming. Metagenomics, that is the gut microbiota and the study of the genome of microbiome. Epigenomics, pharmacogenomics, evolutionary studies, biodiversity assessment. So you can see that the term genomics is very much important for a wide range of applications. And genetics is mainly for understanding hereditary diseases, where certain gene mutations, there are single nucleotide polymorphisms, diagnosing single gene disorders, genetic counseling, breeding studies, all of this comes under genetics. Now we'll move on to the basic DNA and RNA. So we know that there is cell. Cell needs instructions to work, right? So cell needs instruction to build, maintain, respond to environment, and reproduce. And for this, the basic functional aspect that helps in all of this is the nucleic acids. And nucleic acids are DNA and RNA basically. We'll start with DNA, that is deoxyribonucleic acid. So earlier, people used to believe that proteins, they are the hereditary material due to their chemical diversity. And afterwards, in 1928, Griffith's transformation experiments came into the limelight. And he told that there are some transforming principle in the cell, or there is some principle called transforming principle that he named, that could transfer virulence between the bacterial strains. That was in 1928. In 1944, a group of people, Avery, MacLeod, and McCarty, they said that transforming principle of Griffith was actually DNA and not protein, as they earlier thought it to be. And in 1952, Hershey and Chase used bacteriophages labeled with radioactive phosphorus and sulfur and demonstrated that DNA enters the cell during infection. So that is how they came to deduce that DNA carries the genetic information and not the proteins. And in 1953, the decisive year, as we all know, Watson and Crick, along with the backing of the Rosalind Franklin's X-ray diffraction data, proposed the double helix model for DNA. So this is the rough timeline how DNA knowledge came into being. So basically the structure of DNA, it's a polymer made up of repeating units called nucleotides. So if we show it in a very illustrative manner, so we have a sugar at the center. So it is basically a pentose sugar, and I will later show the structure in the side, in the chemical structure. So here, in DNA, the sugar is called deoxyribose. it is connected to a phosphate group, and at the other end, it is connected to a base. So we all have we have all studied in school that there are 4 bases mainly in DNA: A, T, C, and G, adenine, thymine, cytosine, and guanine. So if you look at it structurally, so here, this is the pentose sugar. So if you can see, it has got 5. So all these corners, we call it 1 prime, 2 prime, 3 prime, 4 prime, and 5 prime. So it is to be noted that in a pentose sugar, always at the 1 prime end, the base is connected. In DNA, as I said earlier, the sugar is deoxyribose. So the name comes because at the 2 prime end, there is no OH group. It's only H group. So because it is deoxy, so there is an absence of OH group here at the 2 prime end, because of which it is called deoxyribose. And then at the 5 prime end, it is connected to a phosphate group. So this is the basic structure of DNA. And to join one nucleotide, so basically one nucleotide is joined with another nucleotide. So here, to join one nucleotide with another, the 5 prime phosphate is, so here, this 5 prime phosphate of one group is connected with the 3 carbon of the adjoining nucleotide, linking them together with the phosphodiester bond. As you can see, this is one nucleotide, this is the 5 prime end, and this is the 3 prime end. So every nucleotide, the 3 prime end is connected with the 5 prime end of the next nucleotide with a phosphodiester bond. There are 2 ester bonds that are being formed. So it is called a phosphodiester bond, hence making a full chain of nucleotides. So the 4 nitrogenous bases, so we told about the phosphate and the sugar. Now the 4 nitrogenous bases, it can be classified into 2 groups. One is purine and pyrimidine. So purines are adenine and guanine. They are basically double ring structures. And pyrimidines are single ring structures, cytosine and thymine. So there is Erwin Chargaff in 1950 brought about the Chargaff's rule. That is A equal to T and G equal to C. So basically in a DNA molecule, the amount of adenine always equals the amount of thymine, and the amount of guanine always equals the amount of cytosine. So this rule was foundational to understanding the DNA's double helix structure and its complementary base pairing. So these are the structures of adenine, thymine, guanine, and cytosine. As you can see, adenine is a double ring structure, thymine is single ring, guanine again is double ring, and cytosine is single ring. And these are how the A binds with T and G binds with C. In 1953, James Watson and Francis Crick, guided by Rosalind Franklin's X-ray diffraction data, proposed the famous double helix model. So this is the structure of the DNA in a very 2D perspective. So if you can see, this is one strand of the DNA, as we said earlier, where the nucleotides are being connected. So DNA consists of 2 anti-parallel strands, one running from 5 prime to 3 prime, and the other running from 3 prime to 5 prime. So that's why they are called anti-parallel. The strands are held together by hydrogen bonds between these complementary bases. So as you can see, A and T and G and C, the bonds that are formed between are the hydrogen bonds. So they are held together by hydrogen bonds. A pairs with T via 2 hydrogen bonds and G with C via 3 hydrogen bonds. The 2 strands wind around each other to form a right-handed helix, and each strand serves as a template for the other for replication. And so basically we all know DNA, it is long-term genetic information storage. That is the major function. If you say it in a very gross way, the major function of DNA is for long-term genetic information storage. So this is what we know as I said earlier, chromosome. So we can see that the chromosome, it has chromatin fibers that make up this dense chromosome. And there are structures called histone around which the DNA is bound to form a structure called nucleosome. So the histones and the DNA wound along it basically makes it very compact to be accommodated inside the chromatin, which later makes up the chromosome. And the DNA, as we know, is a double helix. So human cell contains 3.16 billion base pairs of DNA organized into 46 chromosomes within the nucleus, along with There is also mitochondrial DNA other than nuclear DNA, which we will later tell in the later sessions. And if this DNA is uncoiled, the DNA from a single cell would stretch over 6 feet. And the total DNA in the human body could reach over 34 billion miles if lined end to end. So you would have heard it multiple times about the length of the DNA being told to understand the magnanimity of the structure. Next, we will just tell what is the genome size. So while understanding DNA, there are certain terminologies that we use to say the genome size. So basically, two bases when they are connected is called a base pair, right? So the base pair means just one. Two bases connected and base pair is just one. And we tell the genome size in base pair when we are dealing with structures like genes, like a gene having certain sequences. We call it as, okay, it is 50 base pair or 100 base pair or 150 base pair. So that is the base pair. Next is kilobase. So in 1 kilobase, there are 1,000 base pairs together. So in bacterial genes or while explaining about plasmids, we express the genome size in kilobases. Next is MB. MB or MBP. It is megabases. So it's basically bacterial genomes or large genes we express in megabase pair. And GB is the gigabase pair. So basically human genomes are always expressed in gigabases. So all the eukaryotic genomes, not just humans, it is always expressed in gigabase pair. So genome size refers to total amount of DNA contained within one copy of a genome. So it usually measures as the number of base pairs, total count of A, T, G, and C nucleotides, or as the mass of DNA per haploid nucleus, that is N equal to 23, how much is present in 23 chromosomes, and is often expressed in picograms. So this is just the terminology of how the quantification of DNA is told. Next, we'll come to RNA, ribonucleic acid. So if you remember the same structure from the DNA, the only difference is that, main difference is that the pentose sugar here is the ribose sugar. That is, it is not deoxyribose, it is ribose. So we have an OH group at the 2' end of the pentose sugar. It's usually RNAs are single-stranded, not double-stranded like the DNA. And RNAs, this extra hydroxyl group actually makes it more reactive and less stable, and it is only suitable for temporary roles. So DNA is more prone to degradation than RNA is more prone to degradation than DNA. And another major difference between DNA and RNA is that the nitrogenous bases that is involved here uracil is used instead of thymine. So RNA doesn't have a T, it has a uracil, that is U. And rest everything, the functions, the structurally rest everything looks the same. And it carries messages. So RNA has a wide range of functions. So it basically carries messages, acts as an adapter, catalyzes multiple reactions, regulates gene expression, among the few. So before going into the next step, I would just walk you through two major functions, that is transcription and translation. You would have heard of transcription, the method of which DNA is being converted into a messenger RNA is basically called transcription. And messenger RNA later coding into proteins is called translation. So the chart here is basically the codons that are used while coding to amino acids, which later form the proteins. And each amino acid here will have a specific codon that is already given here. So later I'll explain how this structure, this mechanism is being functioned. So basically there are like 4 types of RNA. So we just generally told about the basic structure of RNA. Now coming to different types of RNA. So messenger RNA is the mRNA. So this is what we are seeing here, DNA being transcribed into mRNA. So this mRNA carries the genetic message from the DNA in the nucleus to ribosomes in the cytoplasm. As we know, there is cell, there are multiple cellular organelles, and then there is nucleus, and then there is DNA. So ribosomes are in the cytoplasm, not inside the nucleus. So it is in the cell cytoplasm. So the DNA, the genetic message from the DNA is converted into mRNA. mRNA comes to the cytoplasm to communicate to the ribosome for protein synthesis. So messenger RNA's main function is to aid in protein synthesis. It is a single-stranded molecule with a specific sequence of nucleotides that specifies the amino acid order needed to build a protein. Next, another RNA that is very much, so basically messenger RNA helps in the, is basically playing a role in the transcription phase of the protein synthesis. Second is transfer RNA, tRNA. It is an adapter molecule that facilitates the protein synthesis by delivering the specific amino acids to the ribosome, where they are added to a growing polypeptide chain during translation. So tRNA basically helps in the translation phase of the protein synthesis. So I've not added the structure of tRNA here. So the tRNA basically, after the messenger RNA comes to the ribosome, these specific codons in the mRNA are being recognized by complementary anticodons in the tRNA, and then it will just make a polypeptide chain of the amino acid that it identifies, and then later this amino acid chain then later goes for folding into the protein. So this is how a protein synthesis happens in the body. Other than messenger RNA and transfer RNA, we have got ribosomal RNA. So that is rRNA. It is a major component of the ribosomes, the cellular machinery that synthesizes proteins. It provides structural scaffolding for ribosomal proteins, binds mRNA, attracts the tRNA, and catalyzes the formation of peptide bonds to fill the protein. So basically, ribosomal RNA is the one that oversees all this entire process of mRNA and tRNA. Other than that, there are multiple other RNAs that are regulatory in nature. That is microRNA, small interfering RNA, long non-coding RNA, small nuclear RNA, and small nuclear RNA. So the functions of these will be later explained in the later sessions as to what their roles are in the basic regulation. So after explaining the basic structure of DNA and the organization of DNA and the basic structure of RNA, types of RNA, and the basic function of RNA, we are coming to the central dogma. So dogma is something that is a very, which is the set rule. Okay, so central dogma of molecular biology was proposed by Francis Crick, and it's a foundational concept that describes the flow of genetic information from DNA to RNA to protein. So earlier it was said to be very linear, and any other exception was not considered here. So information is first stored in DNA, and when replication happens, DNA only gets converted into DNA. Then when transcription happens, DNA gets converted into RNA, and RNA, while translation, gets converted into protein. So only this unidirectional flow of information is only to be happening. No other like RNA. It was earlier believed that only this direction is information. This information flow only happens in this direction. So this was kind of the central dogma for multiple years till some exceptions happened. Like there is one reverse transcription. So basically, when they studied some RNA viruses called retroviruses, we all know what retroviruses are. So when this RNA viruses were studied, they found out that these RNA viruses could synthesize DNA from an RNA template using the enzyme reverse transcriptase. So that became an exception to the central dogma that RNA can also be converted into a DNA, because earlier it was told that only DNA, due to replication, can end up producing a DNA. Nothing else can produce a DNA. But this became an exception that certain retroviruses which contain RNA, they can synthesize DNA from an RNA template. So this became the first exception. Second exception is the RNA replication. Certain virus species are so primitive that they use only RNA to directly convert into protein. So this basic thing that DNA converts to RNA, converts to protein is not acceptable here because they don't have a DNA. So it is just directly, they have RNA in their body and it directly transforms into proteins. So basically they don't have a DNA, and that is one another exception to this central dogma. Next is the non-coding RNAs. So not all RNA is translated into proteins. So it is not that the entirety of the RNA in the body is going to be translated into proteins. Many RNA called tRNA, rRNA, microRNA, siRNA, as I explained in the earlier slide, they have regulatory function. As I said, tRNA helps to produce amino acids. mRNA helps carry the information from the nucleus into the cytoplasm. So these RNAs doesn't have a protein directly translated into proteins. So these are called non-coding RNAs. So that again became an exception that all RNA is not translated into proteins. Next are the prions. Prions are infectious protein particles that were later discovered, and they can induce misfolding of normal proteins. So the prions kind of forces a protein to change structure and later becomes misfolded and becomes some other form. So, and that later causes multiple diseases. So this is kind of a protein inducing change in another protein. So it's like a protein makes another protein kind of a pathway. And that is also another exception to the central dogma that only RNA can lead to production of a protein. So this is the central dogma and the exceptions as it is right now. So this is later Francis Crick also accepted that it was not actually unidirectional, and they accepted this that it should not, the central dogma should never be unidirectional. All these are also possible. So after DNA, RNA, transcription, translation, we are coming to the major functional unit of heredity called the genes. So genes are specific sequences in the DNA. It's a segment of DNA that contains instructions necessary to produce a functional product, usually a protein, but sometimes an RNA molecule also. Genes are the fundamental units of heredity. They are transmitted from parents to offspring and determine both the structure and function of the cell. So basically, if you look at a gene, these are the basic structures that you can find in a gene. So so basically DNA has got two strands, and while the transcription process, the two DNA strands are separated or prized open by the RNA polymerase enzyme that will come and prize open both the DNA strands. So this is one strand. So it is from 5' to 3' end. It will initially have a section called the promoter region. So these are all basically sequences. The colored area basically translate to multiple sequences like A, T, G, C, like that. So first area is the promoter region. And this promoter region is basically the control panel of the gene. It lies in the 5' side. So this 5' side of the gene is called as the upstream side, and the 3' side of the gene is called as the downstream side. So promoter is the initial sequence. It's a coding sequence, the site where RNA polymerase and transcription factors bind to initiate the transcription. So this is from where the transcription starts. Basically, this is the sequence in the DNA, and while it is being transcribed into RNA, this is where the transcription process basically starts. Next region is the 5' UTR region. That is the 5' untranslated region. So this is the stretch of mRNA between the promoter and the start codon. So this it is basically transcribed into mRNA, but it is not translated, which means this area when this 5', so basically it starts, this is the gene, the transcription starts from the promoter, it identifies the promoter sequence, and then it starts transcribing into a mRNA. So in the mRNA, this 5' UTR region will be transcribed. But later, when during the translation process, this 5' UTR region doesn't play any role because this is untranslatable region. So just like that, in the end also, there is 3' UTR region. During mRNA production, this 3' UTR region will not be translated. Third is the coding region, that is the exons and the introns. So introns are the non-coding sequences of a gene that interrupt the coding sequence. So basically, I can show it here in a better way. So after the 5' UTR, there are exons and introns. So basically, exons are the sequences that directly code for a protein, and introns in between are non-coding sequences. So basically, during transcription, this is the gene, transcription happens, pre-mRNA is produced, and then later, while making the proper mRNA, these introns are spliced out. So basically, removal of these sections of introns, because they don't generally code for a protein. So these introns are removed, and this removal of these introns are called as RNA splicing. So they are transcribed into pre-mRNA, but later removed during a process called splicing. And exons are the coding segments that remain after splicing and joined together to form the mature mRNA that carries the correct genetic information for protein synthesis. Then, as I said earlier, next comes the 3' UTR region. And after that, so this is the 3' UTR region, and it is transcribed but not translated again, and it plays crucial role in regulating the mRNA stability, localization, and translation. So this 3' UTR region here, while it is being transcribed into mRNA, adds a poly-AA tail. So poly-AA tail means a sequence of adenines that get added here to make it stable. So basically, I told earlier that RNAs are not stable. So to make it more stable, this 3' UTR region is responsible for adding a poly-A tail here so that the mRNA that is being produced here remains relatively stable. And other than all of these, there are multiple other regulatory elements like genes that are regulated by non-coding DNA sequences. So they are not present in the genes. These regulatory elements might be present somewhere else in the DNA, but they have a direct impact on the gene itself, like enhancers, silencers, insulators. So enhancers enhance the transcription and translation. Silencers kind of reduce it or kind of stops it. Insulators are the like there are multiple functions like that, but these are not present on this specific gene. This might be present on some other sequence, but they have a remote effect on the transcription translation process. So to summarize, in the DNA, there are specific gene sequences. It starts with the promoter region. Then there is a 5' UTR region. Then there is exons that directly translate to proteins. There are non-coding areas called as introns. And it ends with a 3' UTR region and like there's a stop codon and the 3' UTR region. While transcription, the 5' UTR region is retained. Exons and introns after splicing becomes introns are removed and only the exons become the protein coding region. And with the poly-A tail, the 3' UTR region results in the addition of a poly-A tail at the end so that this mRNA is relatively stable. After all of this happens in the nucleus. After this, the mRNA is transferred out of the nucleus to the ribosome for the further translation process. So this is the basic structure of the gene and how it helps in the transcription and the translation. So now we told that here during splicing, all these introns are removed. Now there is another phenomenon called alternative splicing. So again, I'm repeating, there is DNA, there is exon 1, exon 2, exon 3, exon 4, and exon 5. And in between, there are introns which will be later spliced. So in the initial example, I told DNA is converted into mRNA. And in this mRNA, all these introns will be spliced. And this is the mRNA that we get according to the first example, having the exon 1, exon 2, exon 3, exon 4, and 5 linearly, they are joined together. But alternative splicing is a form where certain introns or certain exons can be like spliced off selectively. So if here the exon 3 is not added. So if the exon 3 is selectively spliced, only 1, 2, 4, and 5 can be joined together, and this codes for another protein. So this mRNA will code for one protein isoform. This will code for another protein isoform. And here, when the exon 4 is being spliced off, it gives another protein isoform having only 1, 2, 3, and 5. So basically, one gene can actually code for multiple proteins. That is why so many proteins can be coded. So basically, this is what is one of the major functions in our genomics, if you see, because this one gene coding for multiple proteins actually increases the functional aspects of our body, because there are multiple proteins that are helpful for multiple functions of the body. And this kind of condenses the multiple functions within one gene itself. So I hope this alternative splicing is clear. Now comes the basic. So these are the basic functions of DNA, RNA transcription, translation. So as I said earlier, this codon chart that we have. So basically, I told that the mRNA is being synthesized in the nucleus and then later comes out of the cell to go to the ribosome for protein translation. So there is another structure, as I told earlier, called transfer RNA. So these mRNA here will have different sequences like U, A, G, C, like that in random forms. So basically, sets of three nucleotides are called as codons. So here in the mRNA, first three will be called one codon, second three will be called another codon. So all these codons have specific direct connections, like here U. It is where is it? So here, CCU directly codes for proline amino acid. So basically, UUA codes for leucine amino acid. So basically, all of these sets of three codons that are in the mRNA codes for a specific amino acids. So when it goes to the ribosome, so there is tRNA. tRNA has anticodons in it. So basically, each tRNA has one amino acids attached on top, and it has a specific anticodon that is specific to this amino acid. So as the mRNA reaches the ribosome, the specific anticodons will come and bind to the specific codons here and then release the amino acid attached to the tRNA. So when, let's say, CCU comes and binds to the complementary codon here, leucine gets released, and then next tRNA will come and bind to the specific anticodon, and let's say proline is released, and the released amino acids will be linked together with the polypeptide chain, and hence a long linkage of polypeptide amino acid chain is formed. And this polypeptide amino acid chain is later sent for protein folding. So basically, it is folded into structures called protein, and that is how the basic translation happens. So this is the kind of overview. So all of these, what I have told, will be taken as separate lectures in the coming classes, so that this will be understood much in depth and much in detail with specific examples. Now coming to the next topic, that is the projects that shape the modern genomics. So it has to be started with the first project that made the groundbreaking discovery, that is Human Genome Project. It is launched in 1990. It was launched in 1990 and completed in 2003. So this is the beginning of the genomic era. And the main aim of Human Genome Project was to decode the entire human genome. So as I said earlier, in the 20th century, multiple sequencing techniques were developed. So before that sequencing techniques were developed, they did not have any way to sequence the entire human genome. So once the sequencing techniques like Sanger sequencing, which again you will learn in a separate lecture called genomic technologies, different sequencing techniques. So after the sequencing techniques were invented, this Human Genome Project is one of the major groundbreaking discoveries that happened. So it remains one of the most ambitious scientific discoveries in the history. And the objectives of this Human Genome Project were to identify all the genes estimated like at around 1 lakh and now known to be around earlier people thought that there are like more than 1 to 2 lakhs of genes, that because there are so much of proteins that are coding for different functions. Earlier it was thought that there are multiple, like more than 1 lakh genes. So after this Human Genome Project, when they sequenced the entire human genome, they came to know that genes are only 20,000 to 25,000. And now I think you know the reason why genes are very less. Because as I said in the alternate splicing slide, one gene can code for multiple proteins. So that brings down the number of genes to like almost between 20 to 25,000. So second objective was to determine the complete sequence of the 3 billion base pairs of human DNA, store this massive data in public databases accessible to scientists worldwide, improve sequencing technologies and computational tools, and address ethical, legal, and social issues (ELSI) arising from the genome research. So these are the major objectives of the Human Genome Project. And in this, the global collaboration, multiple institutes had taken part, like the NIH, that is US National Institute of Health, US Department of Energy, Wellcome Trust in the UK, then the Broad Institute, MIT and Harvard, the sequencing facilities there. Then the Washington University Genome Sequencing Center, Max Planck Institute, Germany, Genoscope, France, Riken, Japan, and many other auxiliary institutes also made this project happen. And as you can see, the milestones in 1990, the project was launched officially. In 1995, a proof of concept study was conducted in the first bacterial genome, that is Haemophilus influenzae. In 1999, the first human chromosome, that is chromosome 22, was fully sequenced. In 2000, the draft of the human genome announced jointly by the Human Genome Project and Cellular Genomics, and 2003, completion of the reference human genome, coinciding with the 50th anniversary of DNA double helix discovery. So these are the major key points so what are the outcome of it? So they found out that the genome size is, the human genome has about 3.2 billion base pairs. Gene count was not 1 lakh, it is just 20,000 to 25,000 genes, fewer than expected. Then protein coding DNA, that is, we know that DNA, there are like 3.2 billion base pairs, right? But only 1.5 to 2% of the genome codes for proteins. That was a very groundbreaking discovery because everyone thought that the whole DNA translates itself into proteins. But only after this Human Genome Project came into being, they came to know that this entirety of this massive DNA, only 1.5 to 2% of this genome code for proteins. And the rest are all non-coding DNA. Like over 98% is non-coding, but includes important regulatory elements. So basically, the other 98% of the non-coding DNA are basically important for the regulation of multiple processes of, let's say, transcription, translation, etc. Then another key finding was the genetic variation, that humans share 99.9% identical DNA, and only the remaining 0.1% that is different is what causes the inter-individual variability between people. And then there are repetitive DNA. Nearly half of the genome consists of repetitive sequences. Then chromosome mapping. All 23 chromosomes were sequenced and mapped. High similarity with other species, like 98% of sequence similarity with chimpanzees. And basically, the ethical focus on the genomic research was laid down during the Human Genome Project, like ethical, legal, and social implications of genomics. In the post-Human Genome Project era, there are multiple projects that came into being, some very prominent of which only I'm mentioning. The 1000 Genome Project is one among them, from 2008 to 2015. The main objective was to catalog common genetic variations like the single nucleotide polymorphisms and structural variants in at least 1,000 individuals from diverse populations. This study identified more than like 88 million variants across human populations. Then after this came the ENCODE project, that is from 2003 to present. ENCODE means Encyclopedia of DNA Elements, aimed to identify all the functional elements in the human genome. They discovered that over 80% of the genome is biochemically active, and vast regions previously thought to be junk DNA have regulatory roles. After this comes the Human Microbiome Project. By this time, by 2007, the bacterial DNA, the bacteria that inhabits the human beings and helps in multiple functions of our body. So these microbiomes are also started to be sequenced. And this Human Microbiome Project is one of the major projects that happened in between 2007 to 2016. Explore the genome of microbial communities living in and on the human body, multiple body sites. They discovered that humans harbor trillions of microbes whose collective genes or microbiome outnumber human genes by 10 times, more than 100 times. Then comes the Indian Genome Variation Consortium, IGVC. launched in 2002 by CSIR as a precursor to population scale genomics. This created a map of genetic diversity across Indian populations, considering caste, tribe, and linguistic lineages. And it generated invaluable data for understanding population structure, disease susceptibility, and pharmacogenomics. Uh, After this comes the first Indian genome sequencing in 2009. So, in 2009, the Council of Scientific and Industrial Research, CSIR, and our Institute of Genomics and Integrative Biology, achieved a historic milestone by sequencing the first human genome in India. And it was led by Dr. Samir Brahmachari, the then DG CSIR, and the previous director of IGIV. And next program that in India that is very much highlighted is the Indigen program, 2019 to present, led by again CSIR IJB and CSIR CCMB. This flagship initiative aimed to sequence the whole genomes of thousands of Indians. Uh, uh, Yeah, okay, sorry. The Indigen program was led only by CSIR IJB, and this flagship initiative aimed to sequence the whole genomes of thousands of Indians to create a comprehensive Indian reference genome. The phase one was sequenced like around 1,008 genomes are already sequenced by 2019. Now phase two is ongoing, targeting 10,000 genomes with clinical correlations. And the Genome India project, launched by Department of Biotechnology in 2020. It's a national initiative modeled on the Human Genome Project and coordinated by basically DBT and CBR, Center for Brain Research, IISc Bangalore, and involving over 20 national institutions. Objective is to sequence and analyze the genomes of 10,000 Indians across diverse ethnic, linguistic, and geographical backgrounds. And as far as another groundbreaking thing that I have to say is CSR IJB is also working since 2002 on bridging Ayurveda with modern genomics, one of its kind, towards predictive marker discovery for personalized and preventive medicine, and has pioneered the field called Ayurgenomics, in which we are working now. And we have made multiple discoveries and findings. In detail, it will be explained in the session by Dr. Bhavana Parashar herself about Ayurgenomics. And so these are some reading materials that you can go through. These are the first two are major textbooks that are uh, genomics oriented, and the last three, many of you might have heard about it. They are the non-fiction books by different authors: The Gene and Intimate History by Siddhartha Mukherjee, Genome, the Autobiography of Species in 23 Chapters by Matt Ridley, and Double Helix by James Watson. So these you can go through. They're very interesting books, especially the last three. And I think I hope I was able to provide an overview of the genomics, and thank you for patient listening.
Thank you so much, Dr. Sangeeta.
Yeah.
Participants, if they have any doubt, they want to ask anything, they want to clarify anything, they can put their queries in the chat box. Dr. Sangeeta, there is one query from Dr. Samprad. Are you able to see that?
I'm not sure.
Which integrated data bank or resources provide genomics to phenotype datasets, combining variant, gene function, and phenotype level information, which are suitable for translational analysis?
Data banks. So the question actually, I think there are multiple data banks caters to each of those questions that you have asked. That's a very interesting question because we have actually kept one whole lecture for this, exploring different data banks, different resources, how to do literature, basically this genomics to phenotype data, genomics to phenotype data sets. Like for example, I'll give an example like the translational part of it. Like there is a data bank called UniProt. So in the UniProt, it gives the full details about the different proteins of it. Likewise, we have multiple data banks and resources. It will be given in an interactive form in the last lecture. Yeah, okay. Ma'am wants to add something. 1 second, I'll just unmute, ma'am.
Yeah, so that's a good question, and considering this kind of a question which are going to come, and we also wanted to give such resources in a very systematic manner, we have kept one full lecture on that. But to answer the question today, there are several studies conducted called genome-wide association studies, or genetic association of diseases, or field-wide association studies. So the data coming out of these studies have been housed in certain databases and catalogs. So that's called, there's one catalog called Jiva's catalog, which houses all the genetic variation related information and which phenotypic trait or which disease they have been shown to be associated. So that's called Jiva's catalog. Then there is another one called PheGenI, which is phenotype to genotype integration. That data houses all the genetic variation information linked to different kinds of phenotypes or diseases. There are other databases also where clinical condition related variations are collected in one place called as ClinVar. So there are so you can note down for today maybe genome-wide association studies catalog, phenome-wide association study catalog like PheGenI. Then there is a ClinVar database. So these three actually will tell a lot about collection of all those studies and their findings at one place. You can, yeah, and they also give the reference to those original papers and also give more details about which population it was studies, what was the size of the population or the sample size that was taken, what was the significance level, and all that is also given in that catalog. So that's the first thing you can do. And if you want to read more about that SNP per se, as to in gene in which position that SNP is falling, that is given in the DB-SNP. If you type the name of the SNP, single nucleotide polymorphism, which has an ID, each single nucleotide polymorphism has an ID called as RSID. If one types that RSID in the database of SNPs, then you get more detailed genomic information regarding that SNP ID. So this is all in the public domain, and you can read as much as you want in detail from there. About every single gene also, if you want to read that, if this gene is polymorphic and it is giving rise to this disease, why is it so? What is the function of that gene? Then there is a database in the PubMed, it's in the NCBI, you have a column called gene. And in that, if you type the name of the gene, or there's a gene card called website also, if you type the name of the gene, it will give every detail about that gene, where does it express, how does it function, in which cellular localization, in which tissue it is more active. Every information about that gene is available. So gene card for gene, DBSNP for SNP, Jiva's catalog, PheGenI, ClinVar. These are the sources from where you can get more comprehensive information about genes and variations and their association with phenotypes and diseases. Yeah, so UniProt and String are the ones which are talking about internal biological networks of genes. Whereas if you want to study more about its relation with the clinical phenotype, then it is these databases. So I hope I have answered for now, and we will have a full session on this one day. मेरा सुनाई दिया था?
Yes, madam. Yes, madam. So,
Dr.
the part-
Sangeeta has actually written down in the same chat box the names
Yes,
of
yes,
the
yes.
databases.
Yes.
If people want to note down, they can do so.
Are there any other questions about the lecture today? So basically, the lecture today was just to introduce you and make you familiarize with these terms so that when you go into individual sessions, you will be primed better. So we will have classes on genome organized structure and organization, then genome variation, gene expression and regulation, then about this all the databases and all literature search, how to do literature search, and about all the databases, and in an interactive way, like how it will be shown exactly like how to search in each of these databases. And I hope today's session was informative. And if any more doubts, you can write in the chat box.
Participants, if they have any query, they want to clarify anything.
There are multiple YouTube videos also these days where it gives in a very interactive way all of this, like how mRNA comes from nucleus and transports to ribosome. So anyone interested can check that also.
Yeah. Dr. Sangeeta, we have created one Google Classroom for the participants. So if you can uh, give the resources or the YouTube links where they can find all those, so it will be
Yes, yes,
really
sure,
helpful
sure.
for them,
Definitely, yeah.
because this is the very initial exposure to such topics for many of the participants. So more learning resources, some may be
Yeah, more
helpful for them.
audiovisual.
More audio-visual ways of I can include if you can send the link of the Google Classroom, I can add all the resources and the reading materials there.
Okay, sure, we will share.
Basically, the understanding of genomics will all help clinicians in multiple ways. But basically, if anyone is aiming for research or coming into venturing into research field, knowing about what is there and what are the technologies available will always better help in framing your research question and to understand what techniques to use to reach your objectives. So that is
Read the
also,
reports.
and also to understand the current literature in the upcoming studies that are happening. So to understand the reports and research findings. So that is the major objective of the class.
So can we have some questions at the end of each class for the audience? And
They
some
can
of them
answer.
can actually answer also. In the classroom, can we post some questions also?
Yeah, is there a
Yeah.
option like that, sir?
In the Google Classroom
Type
also
them,
we have.
uh,
Here also the participants, they can put the query in the chat box, or in the Google Classroom we have, there we can post also, or we can prior to the session also we can provide some material so that they can read, they can come
yes.
prepare with some of the
Yeah.
things. So that is also possible,
Right,
because
right.
these all are the very new topics which are included in our PG curriculum. So many of the participants, they are not well aware of such things.
You
So
can also take a feedback as to if they want to have it much more slower or after today's class, whether it should be made to go a little slow or maybe, I mean, that is something feedback as an organizer you can take and convey to us so
sure,
that we
madam.
can, yeah. And we would also like to frame some questions for them so that, they would be able to recall what they have read and they would be
Ha,
encouraged to go to the resource material.
yes, madam. In the between PPT, you can post some of the question so that the participants, they can reply in the chat box. That is also possible.
Then from the next lecture, we'll put some questions at the end, so
Yeah.
that from the current lecture, whatever the lecture is, so people can answer in the chat box, and maybe that will be a good way.
So that is possible.
That will also make it more interactive. Okay, if there are no further questions, then we can close the session, sir.
Yes, madam. So thank you so much to Dr. Bhavana Parashar, madam. And thank you so much to Dr. Sangeeta. It
Sure.
was really very informative session. And I request to please share some of the resource material so that we can share with our
Yes.
participants. So next
Definitely.
class they may become with more preparedness.
Okay, sure.
Okay. Thank you so much
Thank
once
you.
again, madam. Thank you so much. So again, for all the participants, tomorrow the session will be there for on this genomic science at sharp 2:30 p.m.
Right, okay. Okay.
Thank you one and
Thank
all.
you.
Thank you so
Thank
much.
you. Right.